I will. I will.
So it's crazy. Don't have one here.
Okay. No, no, I will. I will be below 50 minutes. I prepared like for 40 minutes, so therefore, I think I should be in time.
Give me water, you're French.
No,
Okay, I'm not from Greece, but I can still stuff.
no, you're not stealing. You can have my watch. But the thing is, I will not talk too long because I was only wondering.
This way, I know what's going on. And you'll have that, and you
Yeah.
already know that this is prepared.
Yeah.
That's also for talk, so it's okay.
Okay.
Now all I need to do is to make sure that I forget your watch.
No, no, I will. I trust you. We know each other so long.
Good afternoon. It's a pleasure to introduce my colleague and friend Kamil Uludag. Kamil Uludag pursued his PhD with Arno Willinger at Humboldt University in Berlin, developing methods for NIRS optical imaging, NIRS optical spectroscopy. He then moved on to work with Rick Buxton at University of California. Rick Buxton was here about 3 years ago, if you recall, and Kamil studied with him mechanism of the Bolt signal. He then became a group leader at the Max Planck Institute for Biological Cybernetics in Tübingen in the high field department, and later on associate professor at Maastricht University. Most recently, he moved to Toronto. He has a position, a full professor position at the University of Toronto, and part-time position at this institute. I'm not going to read it, in Korea. Kamil, over the years, investigated mechanism of bold and modeling of bold response, most recently with regard to laminar specific imaging. And he will present his talk about a high resolution anatomical and functional MRI at 7 Tesla.
Thanks, Amir, for the invitation and introduction. Okay, so when we talk about field strength, we have to think what's happening on the physical level. So the first thing to consider is sort of from thermodynamics. We have magnetization of the tissue. So if these are the sketch of magnetic moments of protons, if you apply external field, you have a magnetic moment. And from the Boltzmann equation, we find that the magnetization is proportional to field strength, which means the higher the field strength, the more signal you have. However, because MRI is complicated and there are coil effects and so on, it has been found that actually SNR, at least for standard resolutions, is more than linear. So that's the main reason why people invest money. They want to have more signal. If this is true, then the 7T compared to 3T, we have a factor 4 times more SNR. So that's a good motivation to go to higher field strength. However, MRI has also other physical parameters. The other one is T1. This is from a paper from 2007. T1 as a function of field strength for different tissue types, white matter, gray matter, blood. And the T1 you see is increasing. This is not a trivial result because T1 was expected to actually converge to go to higher field. That it diverges for different tissue types means the T1 contrast increases with field strength. However, that T1 becomes longer is also bad news because it needs more time to recover to the equilibrium magnetization. So we are becoming less time efficient, but at the same time we are increasing anatomic contrast, which can be useful for arterial spin labeling, for time of flight, angiography, and so on. So good and bad news. Then this is from my paper, 2009. This is relaxation rate as a function of field strength for spin echo and gradient echo. It increases with field strength. This again is a good news and bad news. The bad news is the increasing relaxation rate means the signal decays away faster, which means we need faster acquisition. The second thing is because the relaxation rate increases, we have increased functional sensitivity. And for clinicians, the field strength also corresponds to sort of amount of contrast agent applied, means we need less contrast agents in order to have the same effect size, which means for, as you know, there's a debate whether gadolinium is actually safe, and which means if in going to higher field strength, studies have shown you get for half the contrast dose similar or more contrast at 7 Tesla compared to 3 Tesla. So that's good news. But the bad news is we need faster acquisition. So to summarize this part as a sort of a primer for the field strength dependence, we have basic signal noise increase, T1 and T star increase leading to increased contrast. However, effect of these changes depend on the MRI sequence used, which means not for every sequence you will get benefits. For example, for diffusion, the jury is out whether we actually get better signal. I think yes, but there are people who are saying the gradient is more important than the MRI field strength. But there are also other bad news. We have increasing challenges. I will come to this at the end. Image inhomogeneities and the specific absorption rate, so how much some techniques which work at 3 Tesla might be over the threshold for SAR and not be applied at higher field strength. So in my talk, I will cover 3 topics. I will talk about function MRI, I will talk about quantitative anatomy, and I will talk about inhomogeneities if time permits. Function MRI and field strength. So this is from a recent review paper which I have published. If we go to low spatial resolution means image SNR approximately 80 to 100. This is fMRI signal as a function of field strength normalized to 3 Tesla. And this is from the tissue signal. If you go to 3 Tesla, 7 Tesla, we do not get much increase. So the signal amplitude at low spatial resolution from the tissue is actually not increasing with field strength compared to 3 Tesla. However, this is not true if you look at big veins, because the big veins still increase linearly. This is from the tissue. The real benefit, before going to the real benefit, I want to show that is actually still, you get the big vein signal increases, still gives you more functional contrast. This is from a study from Oliver Speck. They did retinotopy at 3 Tesla and 7 Tesla, same statistical threshold, and you see much more activation at 7 Tesla compared to 3 Tesla. So this is a low spatial resolution study. So there is still benefit if you do standard resolution. However, the real benefit goes if you go to higher resolution. And high spatial resolution means image SNR approximately 10, 20, 30. There again normalized to 3 Tesla. If you go to 7 Tesla, we have 5 to 7 times gain for function MRI. And if you know that temporal SNR correlates with the square root, which means we need approximately 30 times more scanning time in order to get the same statistical values at 3 Tesla compared to 7 Tesla, which means the real benefit you get at 7 Tesla if you go to higher spatial resolution. And this was utilized from Maastricht. Federico De Martino did a nice study on the inferior colliculus. From animal experiments, we knew that inferior colliculus has also tonotopy, which means there are neurons specialized to specific tones. We knew this from the auditory cortex in humans, but in animals, we also knew that inferior colliculus has functional specialization organized in tonotopic fashion. Now, if high resolution is the real benefit at 7 Tesla, then this small nuclei can be investigated, and he found tonotopic organization in the inferior colliculus. But we can also go to LGN, superior colliculus, and other structures where high resolution might reveal more functional specialization. The other big topic, which is sort of very, very popular, there are now approximately 30 papers published per year on laminar fMRI. And laminar fMRI means this. So this is from Bruno Weber and Logatitis. This is CSF, this is white matter, and this is cell labeling, cell density, vasculature. And there's a microcircuit model from Douglas Martin. In a very simple words, the input goes to layer 4. From input from another brain area goes to layer 4. Interaction between brain areas are layer 3 and 2, and feedback is layer 5 and 6, two subcortical structures. Obviously, this picture is too simplistic and probably not true for all brain areas, but let's, for the sake of argument, assume this is the case. If this is how it is done, then looking into high resolution spatial profile of functional activation might reveal the information processing in the gray matter. So this is visualized here. Usually we would do electrode measurements with multi contacts. So if you see activation higher in the tissue, we would interpret as cortical input or output, and input from another brain area here, and feedback on output to thalamus here. So let's look at how it would be at 3 Tesla standard resolution. This is approximately, say, 2 to 3 millimeters, because the cortical thickness can be 2 to 4 millimeters usually. So this is approximately the spatial resolution what we would employ at 1.5 and 3 Tesla fMRI. If we go to 7 Tesla and we go to higher resolution, then we can actually start to look into what's happening within the cortical sheath. So that was actually one of the drivers why groups like MGH, Minneapolis, push for higher and higher field strength. Now, 10 years ago, when people were already pitching this idea, it was actually not very clear whether we can get enough function sensitivity. This is now an example which we did: hemifield stimulation, checkerboard, visual cortex, 0.7 millimeter isotropic, TR of 2 seconds. You see in the coronal view, very nice activation pattern. Now, imagine what this means. 0.7 compared to, say, 3 millimeters is approximately 100 times higher resolution. So we have 100 times higher resolution compared to standard resolution, and we still get a very nice function activation within one functional run. So this is quite a robust and significant result, and this is now everybody who has 7T can do this kind of experiments. Now, when people were starting to do laminar fMRI, they realized the spatial profile of the laminar fMRI signal does not look like what you expect from electrophysiology. This is from our old study. This is fMRI signal change. White matter is on the left, CSF is on the right. And what you see is fMRI signal during steady state increase from white matter to CSF. And this is something which is very incongruent to electrophysiological profiles. For example, from one study, now electrophysiologists demonstrate CSF on top and white matter on the bottom. You see a very different spatial profile, very homogeneous spatial profile compared to the fMRI. So the problem was when we measure fMRI, we can actually, it doesn't correspond to anything what we expect from electrophysiology. And this is a very consistent observation. So where does this problem come from? If you think about it, fMRI signal is not a measure of, or is not measuring neuroactivity directly. What we are measuring is deoxygenated blood, which is primagnetic, in the venous structures. And in the tissue, this surface is white matter. You have ascending veins in the tissue. Imagine now, let's say in this layer, we would have activation. Deoxyhemoglobin would change, but the deoxyhemoglobin has to go somewhere, so it goes up and is transported into the pial vein and then is drained away. Which means even if you have only activity in this one single layer, fMRI would detect signal changes also in the other layers. So therefore, it cannot correspond to electrophysiology. However, this problem we can actually describe, and because this is at the end just a draining problem, so mass balance considerations can be employed to describe this. We have just paper impressed in neuroimage describing this. So what we have to, in order to model this, we have to model the deoxygenated hemoglobin transport in the ascending vein. Now, I will not go into the equations, but the equations, even though they might look difficult, they're actually very simple. What they are saying is we have deoxyhemoglobin change and volume change in one layer caused by local neuroactivity, and this changes then the deoxyhemoglobin and volume in the ascending vein, and then it drains up. And then in the next layer, we have again the local neuroactivity changes supplying deoxyhemoglobin changes in the ascending vein, but there's also deoxyhemoglobin coming from the lower layer. Just mass balance. Or in terms of equation, the volume change is how much blood flow goes in from the same layer, from the layer below, and how much blood flow goes out. So we just count how much goes in and how much is going out. So nothing. There's a similar approach from Jakob Heinzle from Klaas Enno-Staffan's group, but I will not go into detail on the comparison. The same we do with deoxyhemoglobin. It looks a little bit more complicated, but there's nothing behind that except mass balance concentration. We have also with the publication, we have published the code, the MATLAB code. Sorry for the open science people, it's in MATLAB and not in Python. So if you want to play around with the model, the code is there, and you can just download and change the parameters and so on and see what's happening. Now with this model at hand, we can actually now model dynamic and steady state. And we can check certain hypotheses. For example, there's the hypothesis if you see a bump in the layer 4, this white matter is CSF. Some people say this is because in layer 4 you have higher vascular density. So it's a vascular hypothesis. Or the other alternative is layer 4 has higher neuroactivity. So we can now insert both hypotheses into the model and see what comes out. And what you see is actually what comes out is the vascular hypothesis does not explain this bump because the bump is too small, which means if in your signal, fMRI's spatial profile, you see a bump, then it's neuronal in origin. We can also look at dynamic properties like time to peak and posthumous undershoot. And we can compare it with experimental data. For example, this is here from my own study published. You see a bump, which means this is then neuroactivity was high in that layer. Or you can compare it with dynamic curves, which have been measured in fMRI in animals, but also because the equations in principle can be also applied to other hemodynamic modalities like optical imaging. You can compare the model predictions with experimental observations. Now, back to our own problem. Why is the fMRI signal so incongruent with the electrophysiology, and can we do something about it? And the model can help with that. Now in this example, what you see is this fMRI signal, white matter to CSF. What we simulate is just let one layer active and see how much is the bold signal in other layers. So if layer 6 is active, you see activation of the bold signal in layer 6, but also some activation layer 5, layer 4, and so on. If layer 5 is active, layer 6 obviously does not have any activity, but the other layers. So we can actually now use this information to calculate what was a true local activation in the fMRI signal. The way we do it, we take our measured fMRI signal. Again, you see the typical increase from white matter to CSF. And we say the real layer 6 activity was the measured one. Layer 5 activity is the measured one minus some effect from layer 6. And we sequentially subtract. If you do this, what you get is actually from this curves, what you get is this is the local activity, which means using this model, we can remove the ascending vein effect and we can get to the local activity, how strong was the neuroactivity on a local level. Another test we did is a paper which has been published just recently, 2 months ago, from Emily Finn in Nature Neuroscience, where they did an experiment, a memory task experiment. I will not go into detail of the task, and measured laminar fMRI in the prefrontal cortex. In addition to the bold signal, what they had is also a vaso signal. The vaso signal is measuring blood volume, which is assumed to be more specific to neuroactivity. So from their blood volume signal, we created the spatial profiles, what are presumably the neuroactivity. Again, CSF is on top, white matter is on the bottom. X-axis is now time. And what they found is during the stimulation condition, there are 2 peaks. And then they had another condition, there's nothing happening. Then in the other condition with A, where action response was needed, there was another peak later on. Now we can insert this neuroactivity profile into our model. And then what you get is the fMRI signal response based on the bold signal. What you see is this one with 2 peaks generates actually an fMRI time course which increased from white matter to CSF, which means the spatial profile what we had here is fully distorted. So if you would do just the gray necker bold, what you would get is actually would be very misleading. But it gets worse. Here we had only activity in the bottom layers. What you see is actually also in this case, the activity in fMRI signal was higher in the superficial layers, which means if you use the standard fMRI approach and you subtract one condition from another condition, the result would be the biggest difference was in the superficial layers, and you would interpret as cognitive feedback. However, if you know the real neuroactivity profile, this was not true. So we actually can say the fMRI signal without any further processing is misleading. Anyway, so we then looked into whether we can invert this, whether we can recover this profile using this signal, using our model. And what you see is this is now adding noise and making voxels. What you see is we can actually generate these 2 peaks, and more importantly, when there was activity in the bottom layer from the bold signal data with model inversion, we can recover the bottom layer activation. This was presented at OHBM, and we are soon submitting a paper on model inversion. To summarize the fMRI part, in gray necker bold signal, we have, if you, to repeat the initial part, for low resolution fMRI, you still get a lot of benefits if you go to 7 Tesla. But the signal increase is mainly due to superficial veins, which means if you do not care about high resolution information, you still get a lot of signal increase in gray necker bold, shown in the example with retinotopy. However, a lot of people are doing laminar fMRI, and there we have the problem that this spatial profile is distorted by ascending veins. So we have unidirectional spatial blurring towards the surface of the cortex. I have shown you a laminar bold model, which can, in principle, model steady state and transients. And we have also shown that this model can be inverted. One sort of message of caution is using difference between conditions is prone to misinterpretation if you do not any further processing. I will now just mention two topics so that I have enough time also for anatomy. I just want to mention if you are planning to do laminar fMRI, there are two things what you should pay attention. One of the biggest problem in laminar fMRI is the registration. So you can, in low resolution fMRI, you can register the functional data to the anatomic data, and if they are a little bit misregistered, there's not a big issue because often people apply spatial smoothing and so on. However, in laminar fMRI, it has to be perfect because you want to identify the gray matter and you want to be identifying all the different layers. So any mistake done in registration will actually counteract your entire study. So, for example, this is a coronal slice. This is MP2 range, contour T1. They look well registered, but if you look here, this is sort of oblique and this is straight, which means laminar fMRI with this kind of registration would not work. One approach which we take is we distort the anatomy, which means we acquire anatomy which has the same distortions as the functional data, and then we get a better registration. So be careful with registration if you do laminar fMRI. Another study which I don't have time to explain any further, this published in 2018. Usually we do isotropic resolution 0.7, for example, isotropic. However, if you are interested in one direction, say laminar direction, you can actually sacrifice the other dimensions and get higher resolution in one direction. So what we did is 100 micrometer in one direction, and the other direction is 2 millimeter and 1.4. We can actually go, let's say if you would do 0.2 millimeter, we could do 1.1, 1.0 times 1.0 times 0.2, and we can get, if you have 3 millimeters cortical thickness, we have 15 voxels covering the laminar profile, so we can actually get very good resolution. And we have published this approach. Right now, this approach is sort of not fully applicable because this coverage is not very high. But if you are interested in one dimension only, laminar dimension, this is an approach to be considered. Anyway, I move on to the quantitative anatomy. Before moving on, anybody has any question to fMRI at high field strength? So in principle, in the model, you have blood volume as a baseline blood volume as parameter. And actually, the increase from white matter to CSF is due to that the blood volume from the veins increase from the closer you go to the surface. So without it, yeah, so in the model, that's just one parameter. So you can actually therefore say, we increase the parameter, and one of the results I have shown was actually assuming layer 4 was higher vascular density than the surrounding layers. So from the model point of view, is very, in a forward way, that's just a single parameter. The question, more interesting question or difficult question is, if you do not know how robust is your model inversion when you don't know where the highest vascular density is and how high it is compared to the surrounding. So that's, from model inversion point of view, that's a problem, but for forward modeling, this is just a parameter you can play with. Yes?
Can you just clarify, are you saying that the Finn findings from the Nature Neuroscience paper are potentially spurious because they don't account for the...
No, they had the vaso. He was asking whether the Emile Finn interpretation of the data was perhaps erroneous. And it's not, because they had the vaso data, and they have based their interpretation on the vaso, but because they have provided the bold data, you could show that if they would have based their interpretation on the bold data, it would have been wrong. So vaso is in that sense superior, and for us, for me as developing the model was sort of a reference data to test the model. Whether actually, because there was nothing in the model a priori which we tuned to fit that data. It was just assuming their neuroactivity profile was generating what they observe in the bolt signal. So therefore it was consistent. But if you would base only your interpretation on the bolt data, there might be problems. Yeah. If you do not do any spatial bias correction.
But if you apply your correction, then you can just analyze the
Yeah.
Y signal as opposed to.
Yeah, actually, I got sort of more data from Renzo Huber, Veso and Bolt in Somat Sensory. So far, everything is consistent. So the model seems to work, but I would not claim this, that it's now the solution until there are more tests. But I think what certainly you can argue is without any processing, Bolt may be deceptive. Yes? So if you ask Renzo, he would say yes. The reason why so there are a disadvantage to Veso. For example, spatial coverage is not as good as the bolt signal. SNI is not as good. TI is not as good. So there are a lot of disadvantages of Veso, and also you need a lot of tweaking to make it work. Whereas Great Echo Bolt is the simplest method with highest SNR and highest coverage. So you have to then say, if I know the spatial profile is not correct, but I get these other advantages, is it worth it or not? And this is sort of a decision. I think Great Echo Bolt is the way to go, but I'm not dismissing Veso, so therefore I'm actually sort of collaborating with Renzo, and the sequence what he used at NIH was actually from my lab. So, but I still would say, if you can make the Great Echo Bolt work, you get the advantages if you are able to address the disadvantages. So, but it's up sort of to everybody. Yeah. Yeah, I'm more the high spatial resolution camp, but for example, for model inversion, to have the entire time course sampled finely is important. So therefore, as long as the temporal resolution is not too bad, I think spatial resolution is more important, at least from my point of view. But again, the people can have good arguments for the other view. Yeah, I have to say sort of a correction. I call this spatial deconvolution, but I regret it because it's not a deconvolution operator. It's a sequential subtraction, but still you have obviously the noise problem, because if the layer 6 is, for example, wrongly determined, that everything else is propagates to all the other layers. So you have to have good enough data, and if the data is bad, then you cannot do anything about it. The issue is there are some uncertainties because of the model, because you have to make some assumptions, say, blood volume distribution and so on. So if the effect size is small between two conditions, I would anyway not trust laminar fMRI. So that's, for me, I'm more on the critical side, that I say you have to have robust effects, not only to do the model sort of deconvolution, but in general to do laminar fMRI. Yeah. I think it's a good approach, but it addresses a different question, which I didn't talk about, the effect of pile vessels on the tissue signal. And for that, we don't have a solution yet. And for example, what you mentioned might be a good approach. This venous signal, what I'm talking about, the ascending veins, they are very small vessels. So you will not be able to regress them out. But the superficial vessels, they are another problem which need to be addressed. And the approach, what you mentioned, is one of the most promising approaches. Okay, I am now changed topic to anatomy. So we have done few studies on anatomy. The one few studies on deep brain stimulation. So because I was approached by deep brain stimulation people, because they said perhaps we can use 7T to identify subthalamic nucleus, which is one of their main targets. This is not from our study, it's from Korea. If you look at 1.5 Tesla coronal images, it looks all noisy and blurry. At 3 Teslas already looks good. This is the subthalamic nucleus. This is the substantia nigra, globus pallidus, and so on. And you see subthalamic nucleus becomes even sharper if you go to 7 Tesla. So what we did is we identified with 7 Tesla the subthalamic nucleus, and we used the method of Tim Behrens using tractography, because in subthalamic nucleus there have been hypothesized that it has either 2 or 3 distinctions, and part of the subthalamic nucleus are supposed to be limbic, as a part of it is the motor, and the people who want to stick electrodes for deep brain stimulation are targeting the motor part of the subthalamic nucleus. Because it's a very tiny structure, it's very easy to miss. So what we did is we identify subthalamic nucleus, the tractography. Here are the parameters for the tractography. Here you see sort of 6 subthalamic nuclei, and you see the size is very different. So using a template atlas-based approach might not fit every subject very well. So the second, in color code, the blue one is a target of the somatosensory motor cortex, yellow associate cortex, green and the prefrontal, and red the limbic. And what you see is also the parts which are more motor are changing in size and shape. So now we are doing a retrospect. We are not using it for pre-surgical planning, because in order to do this for pre-surgical planning, you have to be distortion free and so on. So there are other problems to be solved. But we are doing a retrospective study looking into the clinical outcome and the electrode localization. So we have published 2 papers on this issue. Now, the other thing, what we have done is we have developed a quantitative MRI protocol for patients where we were constrained to less than 1 hour. And we acquired in this 1 hour MP2 range for quantitative T1, multi-echo gradient echo for T2 star and susceptibility imaging, ASL, blood flow, and resting state and movie fMRI. For MP2 range, we also acquired a so-called SA2 range, because it turned out that the MP2 range, even though it's supposed to be quantitative T1, is not actually giving right numbers in T1 in certain brain areas, and you have to do some correction, and this correction is done using another sequence, SA2 range, which is actually quite quick, approximately 1 minute, so therefore it can be combined with MP2 range. And so the resolution was MP2 range and multi-echo gradient echo 0.7 millimeters, ASL, because it has lower SNR, 2.8 millimeters, and resting state 1.4 millimeter isotropic. Here you see example images. So gradient echo is here, MP2 range is here, so you get T1 map, B1 map. With the T2 star, you do mono-exponential fit, you get T2 star map, and you can also, including the phase, do QSM. With the blood flow, you can get ASL, and the blood flow at 7 Tesla is, we have published a paper on this, comparing 3 Tesla and 7 Tesla with blood flow, but I will not go into the technical details of how to acquire ASL at 7 Tesla. Here you see again example that contrast matters. So for R1, so opposite of T1, you see certain structures in the subcortical subcortex, for example, nucleus caudate, putamen, but all of is equally gray. If you go to T2 star, you can see more structures like globus pallidus, a red nucleus. If you do QSM, you can even see more distinctions. For example, if you look here, you see the globus pallidus interna and externa very well delineated, even though it's very fine delineation. So QSM in subcortical structures is actually very valuable to distinguish all the different subcortical structures. This also we have published on this. Here's again axial slice, coronal slice, and the Q was our target for deep brain stimulation, which you can identify very well. We also did some surface reconstruction. We have done a study on MRI contrast at 7 Tesla, how they are reproducible, and we applied it to MELAS patients, which is a mitochondrial disease. This is in this paper. Here is illustrated the surface reconstruction of the different contrast, cortical thickness, extract from T1, T2 star, CBF. Now I want to point out to one observation. So T1 and T2 star, they look quite similar. For example, the somatosensory motor cortex are delineated in both. However, if you go to blood flow, for example, in the visual cortex, blood flow is high, but also in default mode network, blood flow is high. Whereas in T1 is low in the visual cortex, but in the default mode network is high, which means adding blood flow provides additional information on cortical parcellation. So this is an ongoing study. We have not published this yet, but we have approximately 150 data sets, including on diabetes patients and metabolic syndrome patients, where we investigate whether parcellation using T1, T2 star, and blood flow can add something to this parcellation. And I will also point out that actually cortical thickness is also a relevant parameter, which is not investigated much, but this you get from T1, and as you can see, it's not necessarily agreeing with T1. So therefore, cortical thickness is an important parameter which you should also investigate in addition to blood flow. I think I skipped this one. This is the MELAS patients because my time is running out. So this is from the study, MELAS study published last year, where we looked into the quantitative T1, quantitative T2 star, and blood flow distribution in control subjects versus MELAS patients. And we observed some, one of the main results was the control subjects were actually similar to high mutation load MELAS patients than the low mutation load MELAS patients, which means it's good to not only compare the sort of healthy versus diseased, but also within the disease look at sort of a continuous variables, because there might be compensatory mechanisms in play which might actually make the severe disease more similar to the control subject than the lightly diseased. Okay. Another thing, what sequence which we have developed was aiming at locus coeruleus at 7 Tesla. Locus coeruleus is a very tiny structure. You can barely see it here. That's in the brainstem, and it's very small. It's 2 millimeters thick and 50 millimeters long. And this is a prime example why 7 Tesla might be very useful, because in order to visualize this, you have to go to high resolution. And in high resolution at 3 Tesla, you might reach the limits of SNR. And locus coeruleus has been implicated in many diseases, Alzheimer's, Parkinson, and other diseases. Now, we did the comparison, this published last year, 3 Tesla and 7 Tesla. This is the turbus spinaco, sorry for the small labels. You see these 2 small points is the locus coeruleus. If you do turbus spinaco at 7 Tesla, it doesn't work very well. The reason is it's very difficult to get turbus spinaco working at 7 Tesla. However, we developed a magnetization transfer acquisition, which would actually delineate the locus coeruleus much better. And the acquisition time was actually shorter. This is here 9 minutes compared to 11 minutes. And it had 0.4 millimeter isotropic resolution approximately, whereas this one had 0.4 in plane and 2 millimeters. So we had 4 times higher resolution in a shorter acquisition duration. The locus coeruleus is visualized in the coronal view better. So you see here at 3 Tesla, you can barely see the locus coeruleus, whereas at 7 Tesla, with our approach, the magnetization transfer approach, you see locus coeruleus much better. So this is, and now we are looking into whether health subjects versus Alzheimer patients show volumetric differences in locus coeruleus. So questions to the anatomy before I finish the part with the inhomogeneities. Any questions to anatomy at 7 Tesla? Okay, everybody's, yeah.
Can you explain how is the SA2 MPRH correct the MP2 rate?
So MP2 rate assumes measures sort of at 2 inversion times, and then if it's a T1 recovery curve, you fit the curve or lookup table and you get T1 values. However, this assumes that the flip angle is homogeneous at 7 Tesla, and this is actually the topic in the inhomogeneities. The flip angle at 7 Tesla is not homogeneous, which means for sequences which are not flip angle sensitive, like gradient echo or less sensitive, is not a big problem. But for sequences which are flip angle sensitive, like spin echo or turbo spin echo, as you have seen in the example, 7 Tesla needs optimization. So you need to optimize for region of interest or make different RF pulses and so on. So 7 Tesla, the need for development is to deal with inhomogeneities. That's the biggest problem at 7 Tesla, I would say. Now, if in MP2 range we have a inhomogeneous flip angle and it makes a lookup table to recover T1 curves, then you get wrong values where the flip angle is not good. So SA2 range actually determines sort of the field map, the B1 map, and then it then corrects the lookup table and gives the correct values. And we have investigated this. We have published a paper 3 years ago where we looked at the T1 before and after, and actually also gray and white matter contrast increases if you do the correction. So in certain brain areas where you have not good B1 plus, you have to do SA2 range, otherwise you don't get good gray matter, white matter contrast, and you get wrong T1 values. So it's a known issue. It's necessary to do. So it's not difficult, but a lot of people do not do this. This problem is already true at 3 Tesla, by the way. Only at 3 Tesla is less visible. So therefore, T1 at 3 Tesla might also not be accurate everywhere. Only the effect size of the misestimation is less pronounced at 3 Tesla than compared to 7 Tesla. Yes?
For the locus coeruleus imaging, did you make any modifications to the MT pulse to avoid SAR issues?
Yeah, so it's actually it's a MT turbo flash acquisition, so it's a very specific MT pulse and very specific readout. And with that, we avoid the issues, yeah. So you cannot just use the standard MT acquisition, but and the source of it is not clear yet. Why? Because with standard MT, we also had the standard MT, you don't see the locus coeruleus as well. So it seems to be a mixture of different effects, where we luckily hit sort of the sweet spot where the LC contrast was highest, but it's not clear where the contrast comes from.
So essentially, you're imaging the neuromelanin. Or do you also
That's the debate. It's right now, people are debating where the contrast is coming from. Yeah. So there are a lot of groups are active in that field, and so far it's not clear. Neuromelanin is certainly a contributor, but it's not the only source.
And is the substantia nigra pars compacta also in your field of view? Like,
No, in this one not. No,
it's not.
In this one not. We don't have, because if you focus on that and with that high resolution, the coverage is not very high. Any other question? So I come now to the not good parts of 7 Tesla inhomogeneities. So I want to just show you a standard image. What you see if you do, for example, T1 with MP2 range, you see here the anterior temporal lobe you don't see gray and white matter contrast. And there's low-tech approach which can be helpful. That's called dielectric pads. These are just some substances. This costs like $50, and you put it in plastic bag. If you put it then on the sides, what you get already, it's a very high improvement. For example, here, the problem is not completely solved, but it's obviously you cover much of the signal. So it means you invest millions in the 7 Tesla, and then $50 more, and then you get a lot of signal improvement.
Is the upper image obtained with one channel for transmit?
Yeah, both are one channel transmit. So you use the standard MP2 range, push button, and then you see sort of image imperfections in the anterior temporal lobe, and here, for example, in the cerebellum. So cerebellum does not look like cerebellum. But if you put the dielectric pads behind the back, again, the problem is not solved, but is improved. And in addition, what we did is here we used so-called adiabatic pulses, which also give additional improvement. So these are sort of, there are certain brain areas. Here, for example, if you look at top, everything looks perfect. But if you look at cerebellum, and there's the B1 plus is so bad that you don't get much contrast anymore. Another approach is now 9.4, but it works similarly at 7 Tesla. Again, if you use a standard approach, you get also image inhomogeneities like black spots. If you use KT points, which is an advanced imaging approach, again, you can get the image homogeneity much better. So the short message of this is at 7 Tesla, certain brain areas like visual cortex or smart sensory motor cortex, you don't have to do much to get good signal. But in certain brain areas, the signal quality is not very good, especially the more down you go, because Nova Medical Coil, the transmit efficiency is very low. And in order to get this better, you have to do some tricks. RF pulses, new sequences, or the dielectric pads. As I said, it's very simple to do. And especially if you're interested in these areas, you have to use it. For example, for ASL, we put dielectric pads here at the neck area in order to get the labeling high. Without this, our labeling efficiency was too low, and we don't get much ASL signal. So this low-tech solutions might already reduce the problem of image inhomogeneity, but this is one of the biggest issues. As I said, some sequences like gradient echo are less sensitive to this, but some advanced sequences like turbospin echo and so on are very sensitive to this. To summarize this part, so or in general, summarize 7 Tesla. So improvements of image quality depends on MR sequence. So not everything will be better at 7 Tesla. There are certain things like diffusion where the jury is still out whether 7 Tesla is better at 3 Tesla compared to 3 Tesla, because often, for example, in diffusion, people use spin echo approaches, and if spin echo does not work well, then diffusion at 7 Tesla might also not work well. However, I think, especially if you go to high resolution, for most sequences, 7 Tesla is better than 1.5 and 3 Tesla, and the argument is very simple. If you go to higher resolution, you push the image SNR to such a low level, you don't detect anything, or you don't detect much. So if you have any signal to play with, you're already better. So therefore, at 7 Tesla, going to higher resolution will outperform in most techniques compared to 3 Tesla and 1.5 Tesla. Already at standard resolution, some of the approaches are better, but at high resolution, there's, I think, no doubt in my mind that 7 Tesla is superior. I have not talked too deeply on the quantitative approach, but I think a lot of approaches we have to now be more quantitative, which means quantitative in the sense of physical parameters like quantitative T1 in milliseconds, T2 star in milliseconds, because, for example, some of the comparison we did in MELAS patients was you could only discover the difference between MELAS patients and health subjects based on the quantitative values, because the quantitative values gives you additional information in addition to atrophy and lesions. So if the brain looks normal, however, there are global changes on a microscopic level, they might affect the quantitative values without showing atrophy and lesions. So quantitative values, I think, are very important and provide information MRI, which is usually not acquired or considered. And right now, the techniques have developed so much that actually the additional scanning time increase is not that significant anymore. I think in earlier generations where if you want to do quantitative T1, you are doubling the scanning time, but now it's only like 10-20% longer scanning duration. So actually the price to pay to go quantitative is not that much anymore. And at the end, I very shortly talked about challenges. One of the main challenges is inhomogeneities, but what also you have mentioned, specific absorption rate for magnetization transfer approaches, for turbospin echo approaches, they are still a problem. So some sequence which might work at 3 Tesla might have to be adapted and changed in order to stay within the limits. Then I thank my former group in Maastricht. And if you want to read a little more detail, I edited with John Polimeni a neuroimage special issue last year, neuroimaging with ultra-high field MRI, present and future. In that special issue, you find some technical papers, but also papers on clinical applications of 7 Tesla and cognitive applications. So there's a wide range of papers covering I would say, most of the aspects of neuroimaging at ultra-high field. So it's probably a good starting point if you want to look into more into this issue. And thank you for your attention. Any questions to any part, to fMRI, to anatomy, inhomogeneities?
Very nice talk. In the modeling that you're doing at the beginning with the bold, what's the relative role of the intra and extravascular bold signal do you do?
So at 7 Tesla, at Graydon Echo, most of the signal is extravascular, but we have our bold signal model where we have also the small contribution from the intravascular signal. So we have both contributions, but the majority is anyway extravascular.
What's the typical echo time or range of echo times that you're using?
So in the model, we use sort of the T equals T2 star.
Sorry?
T equals T2 star, which is like, say, around 20, or a little bit more than, less, yeah, around 20 milliseconds. But in principle, this is a parameter which can be varied. Yeah. So in the paper, we actually provide a bold signal derivation, the equation for the bold signal. So if you would have different echo time, you could just calculate it differently. Yes?
Can you speak to the issue of subject motion and that impact when you go from 3 to 7T?
So subject motion, you mean, yeah. So subject motion, I have to say, there were many surprises when I started to do 7T. And at least for the population we were using, subject motion was not a big issue. So even for laminar fMRI, where we have 0.7 millimeter, subject motion was correctable. So I'm sort of still shocked that a healthy human subject within 10 minutes moves 1 or 2 millimeters. But this is, so subject motion is not a big issue. I would not do laminar fMRI probably, not standard laminar fMRI in patients or children, because there you cannot control it that much. But in health subject, it was not a problem. And in the anatomic data, where we had 0.7 millimeter isotropic, say, NP2 range, we scanned 150 subjects, and these were all elderly. So subject motion, to my surprise, empirically, was not a big issue so far. So it's surprising, but it's just an observation.
Can you use perspective motion correction?
No, we don't do any motion correction. It means it's good to use. I was talking earlier. I'm trying to use sort of navigator-based motion correction even at 3 Tesla, but at 7 Tesla, at 0.7 millimeter in elderly patients, we had, I think from 150 subjects, I think 2 people dropped out or the signal quality was not good enough. So, it's surprising, but this is just what our experience is. Any other question? Yeah. Yeah. So resting state, it means I'm not resting state experts, but I would think if you want to do more finer parcellation, for example, then going to higher resolution also helps resting state. For example, we have set up an fMRI protocol whole brain in the same scanner what you have, 1 millimeter isotropic whole brain, TR of 3 seconds. So you have much more voxels, which then means if you want to then subdivide, say, default mode network and so on, if you have more voxels, it's easier to do because you have more variances in the data which can delineate substructures within already these global structures. So I think, therefore, there might be something, but as I said, I'm not a resting state expert, so therefore, how much practical advantage you get, I don't know, but you get many more voxels with good signal quality. The second question, yeah.
But then, do you lose other areas, like the cerebellum and
Yes, it means if you are interested, it means if you want, if it's not a resolution issue. So it means if you would do a 3 millimeter isotropic at 7 Tesla, cerebellum is bad if you don't do anything. So it's not a resolution issue. So about 1 millimeter, actually, if you go to higher resolutions, for example, susceptibility artifacts become less. So like in prefrontal cortex, orbitofrontal cortex, smaller slice thickness reduces susceptibility artifacts. So it's actually beneficial to go to higher resolution. But certain brain areas in general will be a problem, but this is a problem not only for resting state, but also for task-based fMRI and not often related to spatial resolution. For the second question, for the subcortical, there it depends which subcortical structures you want to look at. So if in the maps I have shown you with the T2 star, so the optimum both signal sensitivity is TE equals T2 star, which means if you have no wide variability of T2 star in the subcortical structures and you are aiming at a specific subcortical structure, you probably have to use a different TE than what you would use if you were to image the cortex. So if you have a specific hypothesis and you say, I want to see activity in putamen, then you have to determine in few pilot subjects the T2 star and then choose your TE accordingly. So I think the main thing to adjust would be TE. And when it comes to SNR, the SNR gain is biggest in the cortex and less in the subcortical structures, but as I have shown you in some of the data, also some very interesting data from MGH, where they have shown that 7 Tesla actually also benefits a lot for subcortical structures. Yeah.
I was wondering, do the patients feel any difference in 7 Tesla compared to 3 Tesla, like dizziness, or is there more chances for peripheral nerve stimulation?
Yeah, so the dizziness issue was bigger in the earlier MRI generations because the table movement was constant, and which means the magnetic field was changing from 0 Tesla to 7 Tesla, and the speed of the table movement was irrespective how quickly the magnetic field was changing, and people were getting dizzy. However, now the table speed is adjusted to how quickly the magnetic field is changing, which means the vestibular stimulation is now reduced. So in our study, for example, in the 150 subject study with patients, we didn't have any problem. It means dizziness can occur, but occurs also already at 3 Tesla. So we, in my hands, it was not a major issue. So we had 2 dropouts, but it was not because of dizziness, because of movement, that the data quality was not good enough. And then two others did not participate, but we didn't. And they were scanned at 3T, which means they were comfortable both at 3T and 7T. So, and the second question was?
It's about the peripheral nerve stimulation.
Yeah, the peripheral nerve stimulation is, at the end, the limits is the same at 3 Tesla and 7 Tesla, which is sort of depends on the slew rate. And usually you you can reach that limit easy at 7 Tesla, but the manufacturers prevent that you can code the sequence in a way that you would reach peripheral nerve stimulation. So or in other words, the hardware could do faster imaging and so on, but the manufacturers limit this in order to avoid peripheral nerve stimulation.
Thanks for the great talk. Could you comment something about the impact of physiology on the 7 Tesla bolt fMRI?
It depends on the sequence. So the effect of respiration, like on fMRI signal, is larger because the field fluctuation due to respiration at the head level scales with field strength. So, which means physiological fluctuations have bigger impact at 7 Tesla in general. But there are sequences which are more sensitive to physiological noise than other sequences. So it's really a sequence-specific question, but in general, the impact of physiological sort of confounds are big at 7 Tesla. They got it delivered. So first, I want to see it up the field. So that's a different question. But in general, if it doesn't sound much of a difference, because for example, Minneapolis has 10.5, it's only like 30% increase, or I don't know whether my math is correct. Or, no, it's 50%. It's 50% increase. 50% increase, so it doesn't sound too much. But if effects are, say, quadratic, then already it's quite a lot. Then it's 2 times higher. So it's going to higher resolution. It's also getting more localized signal. Then for image reconstruction, going to higher field is better because the different coil elements are less correlated to each other, which means acceleration and the penalties you are getting with acceleration are less at higher field than compared to 3 Tesla, which means even if you have the same coil at 3 Tesla and accelerate in the same way to get the image acquisition shorter, the penalties you are paying at higher field is less. So there are a lot of advantages going to higher field in addition to the basic signal-to-noise increase. But the difficulties will also be larger, so I don't see a mass market for the 10.5 and 11.7 and so on. But for MR Physics Group, where they have a lot of people working on these issues, they have certainly a good tool to play with. But I think the 7 Tesla is a mature technology, and we had already, after 1 week, very good data. So there's a lot of potential to do a lot of good applied studies. With at 10.5 and 11.7, this will take its time. Yeah, but this is like a cathedral project. Means it will take 10, 15 years until the scanner is there. And as I said, for MR physicists, you have to always distinguish applied scientist and MR physicist, because MR physicists, the problems are their research. Whereas if somebody wants to do cognitive neuroscience, you don't want to have the problem. So therefore, I think the 7 Tesla is now in the sweet spot where the problems are under control. At the same time, there are not too many 7 Teslas worldwide, which means you can do a lot of studies, pioneering studies, with not much competition, also with not too much effort. Therefore, 7 Tesla, I think it's the right time. 10.5 and 11.7, that's on also the 14 Tesla. These are sort of for very specific groups who want to push the limits.
So you mentioned that in your hands, subject motion is not an issue.
Yeah.
And my pose on this is because, I mean, you made this statement, or that's your experience, because you're working with Dutch subjects, and if you have problems, email me because we are developing methods to overcome subject motion.
Yeah, it means no problem. What I meant with no problem is they were still correctable. So it was not that the data quality was so corrupted you could not use it anymore. So in that sense, motion was not a problem. But it doesn't mean that you should not employ clever motion correction algorithms. So especially if you then go to less compliant subjects. Okay, I think if there are no further questions, thank you all for your attention. You didn't steal it. I'm surprised.
I mean, I was